All right, so let's look at blood. Blood is a connective tissue, if you recall. And recall that the three components that make up a connective tissue are a matrix, a formed element, and a fiber. And so the formed elements, as you see here with blood, are leukocytes, erythrocytes, and platelets. The matrix is our plasma. And then the fiber we'll get to in just a little bit. It's fibrinogen. When you do a hematocrit centrifuged, 44% is usually the erythrocytes. 1% is this little buffy coat sitting on top of the erythrocytes that make up the platelets and the leukocytes. And then 55% is our plasma. And plasma is water, plasma protein, small solutes. So erythrocytes have a biconcave shape. So during erythropoiesis, as you'll see in just a few minutes, the erythrocytes spit out their nucleus and become biconcave. And this is to increase the surface area because they're basically just big bags of hemoglobin, because that's their main purpose, is to carry oxygen. And oxygen binds to hemoglobin and is carried throughout the body. Here is the structure of hemoglobin. It's made up of 2 beta chains. So these are proteins. Made up of 2 alpha chains. These are proteins. So this is our quaternary structure. In the middle of each of these proteins, so 4 protein chains, 2 beta, 2 alpha, is our heme group. And within the heme molecule is an iron. And so this little red dot is a molecule of iron, and that's where oxygen actually binds. So oxygen binds here. Majority of our CO2 will actually bind to the globin chains. And so here is the view of the heme in the hemoglobin molecule. And as I said, the center dot is our iron. And so carbon dioxide actually binds to the globins, and so that's why it's called carba for carbon dioxide. Carba amino, binding member amino acids make up our proteins. So carba, amino, hemoglobin, because the carbon dioxide binds to the amino acids of our globin chains. And so about 23% of CO2 is transported in this mechanism. Something like carbon monoxide actually kills people because carbon monoxide has a strong affinity for this iron molecule where the oxygen is supposed to bind. And so the carbon monoxide binds to these sites and prevents oxygen from binding. So basically you suffocate your cells because they can't get oxygen because the carbon monoxide has kicked them off. And so changes in our hemoglobin shape are what stimulate the loading and unloading of oxygen from the hemoglobin, and that is affected by things such as temperature, pH, partial pressure gradients. So how are our erythrocytes and leukocytes, all those formed elements, made? So they are all made with a process, and they all start with this hematopoietic stem cell. So hematopoiesis is the formation in the red bone marrow where all of our formed elements are made. Specifically for erythropoiesis, for making our erythrocytes, we have erythropoiesis. Again, everything starts with the hematopoietic stem cell. Then it starts to differentiate because of growth factors that push it down this pathway. And so the first thing it does is committing to becoming a erythrocyte. Then it continues with the hormone of erythropoietin. And if you recall, erythropoietin is one of those hormones made by the kidney. Pushes it to a pro-erythroblast. Then it becomes an early erythroblast. At this time, your rough endoplasmic reticulum, your Golgi, rapidly synthesizing lots of hemoglobin, because that's what it's eventually going to be, is just a big bag of hemoglobin. So you still have your nucleus, have all of your organelles making lots and lots and lots of hemoglobin. And then once you've made enough hemoglobin, it goes into a late erythroblast. Nucleus shrinks and is ejected with all the other organelles. And then all that remains are all those hemoglobin molecules that were made in our previous stage when we were an early erythroblast. Then it becomes a reticulocyte. Once it gets rid of the organelles, goes into our bone marrow. These are sinusoidal capillaries, so they have large enough gaps to allow cells in and out. And then as it enters the bloodstream, it matures into what we typically think of as a erythrocyte, this biconcave shape. And so what stimulates erythropoiesis? Things like low oxygen levels. So if the oxygen level drops, kidneys make erythropoietin. Erythropoietin goes to the red bone marrow and stimulates erythropoiesis, where we make new erythrocytes. Erythrocytes live about 120 days, and at the end of that 120 days, they need to be broken down. Most of the materials that are in the erythrocyte are reused. Spleen, so this is the spleen sinusoidal, is a red blood cell graveyard. So this is where they really go to die. Come in, you get this macrophage that's in the white pulp of the spleen. It breaks the blood cell down. The hemoglobin is broken into the amino acids and heme. Amino acids go back to the red bone marrow to be reused. The heme is broken down into iron and bilirubin, and iron binds with transferrin, and it has to bind with transferrin because free iron is toxic to us, and so to make us sick. And so it binds to a transport protein called transferrin and is taken back to the red bone marrow to make more hemoglobin. The rest of the heme molecule, bilirubin, is taken to the liver, where it is further broken down and put into our bile salts and secreted out. Is what gives feces its brown color and yellow color to the urine. So it's kind of filtered out of the system or excreted out via the kidneys or the feces. And again, that gives the feces the brown color and urine its yellow color. Okay, we've talked about erythrogranulocytes or leukocytes. We have two types, the granulocytes. We have neutrophils, which are kind of a purple color, 3 to 5 lobes, cytoplasmic granules. And watch the video in the lab practical folder if you need more clarification. And so the neutrophils are our bacterial slayers. So they go and attack and kill invading organisms via chemotaxis, and we'll talk about chemotaxis in a later lecture. And they directly kill bacterial cells specifically, so that's why they're called the bacterial slayers. Also play a role in inflammation. Our eosinophils are these red bilobed molecules. They are phagocytes, usually against parasitic worms or allergic reactions. So eosinophils are activated during allergic reactions. We don't have many parasitic worms here, but. And then basophils, S-shaped, dark, methylene blue chemicals, granule, mediate inflammation. These are our least common. And so the way you can remember who's most common and who's least common is never let monkeys eat bananas. Neutrophils are the most numerous. Lymphocytes are next. Monocytes are after that. Eosinophils are next, and then bananas, basophils are the least numerous of our leukocytes. Okay, so those were our 3 granulocytes. We have 2 agranulocytes, which are our lymphocytes and our monocytes. And we will talk a lot more about lymphocytes when we get to immunity. So this is our second most common, basically a nucleus with a halo of plasma. Makes our antibodies. So you have 2 types, our T cells and our B cells. T cells are hand-to-hand combat. B cells are the ones that make our antibodies. And so here, B cells make antibodies, T cells hand-to-hand combat. They directly attack and kill cells that activate them. And our monocytes are other big phagocytic cell. So neutrophils are phagocytic, bacterial slayers, macrophages are just our general phagocytic cells. They're also going to be our antigen-presenting cells, so the ones that actually start our response of the immune system. And here's a better picture of those 2 agranulocytes. Here is leukopoiesis. So again, starts with a hematopoietic stem cell, has growth factors or colony stimulating factors that push them either into the myeloid or lymphoid line. Out of the myeloid cell, you get all of the granulocytes and our monocytes. Out of the lymphoid line, you get the 2 lymphocytes. And so you go to the committed cells, which are the myoblast, the monoblast, the lymphoblast, then our precursor cells, promyelocyte, promonocyte, prolymphocyte, and then into the neutrophil band cell, eosinophil band cell, basophil band cell, T lymphocyte, B lymphocyte, and then forming our mature cells. So they all come from that hematopoietic stem cell and go down their respective lineages. Platelets is the last of our formed units, and platelets are actually just fragments of cells. They are anucleated also, and they play a major role in clotting. So platelets are clotting, again, derived from the hematopoietic stem cell, myeloid cell line to a megakaryoblast, and the megakaryoblast, repeated mitosis within cytokinesis gives you a megakaryocyte, and the megakaryocyte just pinches off little pieces of itself with these cytoplasmic arms and puts the platelets, these little flakes. And when you look at a blood smear, they look like little cornflakes, just little pieces of trash throughout the blood smear. But they play a major role in hemostasis, which is our blood clotting. And so these are our 5 steps of hemostasis, how a blood clot is formed. First, at the site of blood loss, where the injury happens, we have vascular spasm to slow the blood flow, platelet plug formation by the platelets, coagulation down our intrinsic and extrinsic pathways, clot retraction, and then thrombolysis. And so here's vascular spasm, cut, blood leaks out, vasoconstriction to stop the blood flow. This is our part 1 of our hemostasis, stopping the blood flow. Then, because the collagen fibers are exposed, it attracts the platelets. The platelets attach to the site of injury where the collagen fibers are exposed. And then they stick to each other and activate and start a chemotaxic attraction. And so the contents of the platelet activate and attract more platelets, so start a positive feedback mechanism and attract more platelets. In addition to forming the platelet plug, the injury starts either our intrinsic or extrinsic pathways, which merge into our common pathway. And this is that fiber element we were talking about. This is where fibrinogen, which is the fiber element of blood, is converted to fibrin by this common pathway. And so what is the common pathway? It's the intrinsic and extrinsic coming together. So here's the intrinsic, starts with factor 12. Extrinsic starts with tissue factor, converts into an active form. So this is inactive, and then when you see the little a, it's activated. And starts a cascade from 9 to 11, and then 5 to activated 5A, joins with 11A. 11A activates 10 to 10A, and then forms with 8, and here 10A, activated by the tissue factor with 7A. And so this is just a whole cascade leading to that common pathway. And so the 5A that we just saw activated in both pathways, right here, 5A, they both activate 5A in the end. The 5A joins with 10A. And so 10A was what was actually activated. And so 5A and 10A then come together along with calcium. These 3 little dots are calcium. And this is our prothrombin activator. So that common pathway ends with the formation of our prothrombin activator. And then the prothrombin activator converts prothrombin to thrombin. And then thrombin converts fibrinogen to fibrin. This is all you need to know, is that the prothrombin activator, which is made as a result of our intrinsic and extrinsic pathways, converts prothrombin to thrombin, and then thrombin converts fibrinogen to fibrin. And then the fibrin is like a glue. It holds the platelets together. And it will also help in clot retraction when that happens. So it's the glue that holds the platelet plug together. And this is that fiber element of our blood that makes it a connective tissue. And so here's a concept boost to summarize what we just went over. Activation of 10A then comes together with 5 and calcium to make our prothrombin activator. That activates prothrombin to thrombin, and then thrombin activates fibrinogen to fibrin. Seals the wound and holds the clot together. All right, once we've got that clot made and stabilized, we're now, all right, with the clot retraction, the platelets squeeze together. They actually have the actin and myosin similar to what we saw with our muscle contraction. And they squeeze, and when they squeeze serum out. And so serum is plasma without any of the clotting factors. So plasma has all the clotting factors. Serum is the plasma without the clotting factors, because they've all just been used to make this clot and to convert fibrinogen to fibrin. And then once the wound starts to heal, it is sealed. Now we need to do thrombolysis, slicing of that thrombus, because when this clot is a thrombus, it needs to be broken down so that it doesn't cause issues when it breaks off. So TPA comes in, activates plasmin, and then plasmin is what actually breaks down the clot. So plasminogen activator, TPA, comes in and converts plasminogen to plasmin, its active form. Then plasmin is able to degrade the clot, and the wound finishes healing. And so here's the big picture of hemostasis and the 5 steps of hemostasis. Here are all those clotting factors that we saw, and again, you only need to know the prothrombin activator converts prothrombin to thrombin, and then thrombin converts fibrinogen to fibrin. But there are over 13 different clotting factors. They're always in our plasma. They're just not activated, and they get activated when the wound starts the pathways. And as we talked about earlier, positive feedback mechanism that is tightly regulated. So this is one of our core principles, and it is tightly regulated by 2 chemicals that regulate the first and second stages of the clot formation. Prostacyclin is a prostaglandin that inhibits platelet aggregation, and nitric oxides will cause vasodilation. And the endothelial cells themselves actually make the anticoagulants antithrombin 2, heparin sulfate, and protein C. And so this is how your body keeps the clot from spreading from the site of injury. And here's a table that shows that. All right, so I think that is it for our blood lecture. If you have questions, be sure to let me know. See you next class.